Okay, so myoblasts are, as I last time I've gone over this a little bit more. Myoblasts are cells determined to produce muscle cells and begin producing muscle-specific proteins. MyoD is a master regulatory gene. So MyoD, you guys, is the name of a gene. When you read your PowerPoints, these at home, and if they don't make sense, just break them down, read them slowly. Encodes a transcription factor that commits the cells to becoming skeletal muscle. MyoD protein can turn some kinds of differentiated cells like fat cells and liver cells into muscle cells as well. So that's what pretty much MyoD is. Here's a nucleus, here's an embryonic precursor, the cells before, and we talked about this. This is a master regulatory gene, the MyoD, we talked about it, and it turns on specific genes, other genes that are off right here. And here, that in this embryonic precursor, that gene is also off. Then that myoblast, that cell becomes myoblast, determined. Then what happens, MyoD protein transcription, this factor is made by mRNA, and then of course, when that is made, the myoblast can become muscle cells, and these are the sequence of events that can become muscle cells. All I'm saying, what this is all about, the body, nuts and shell, what this PowerPoint is all about, and this one and the last one was explaining it. It's saying that a cell eventually, when you have an embryonic cell or a stem cell, if you would, either one of them, when you have a cell, right here, you do have a cell, and inside of the cell, this cell can become other type of cells, skeletal muscle cell, muscle cells, or it can become, I'm just giving you an example, a white blood cell. However, it depends on, this is a stem cell. Okay, however, it depends on what genes are being turned off and on here. For example, on this one, the gene that is making this cell to be turned to a white blood cell is off. The gene that is making the cell, let's say it's a red gene, I'm going to put it up here, I don't have red, green, can you guys see the green? The green gene is off here, okay, because that gene turns this cell, makes this cell to become muscle cell. It's off. However, the gene that is blue, right here, the blue is on, the blue gene is, blue gene, almost like a song. Blue gene is on, and right here it becomes muscle cell. This one, the green gene, right here is on, and the blue gene is off. That's all I'm trying to say. And your textbook is taking one example, that's MyoD gene. We call it MyoD gene, that is being turned off or being turned on. For making muscle cell, it's being turned on. Okay, so that's all that is. Okay, pattern, the next thing that we are going to, the next few PowerPoints is going to talk about the pattern of the cell. How, the question is, how in an embryo, you remember we had it, that was a lab practical question. You had a ball of cells right here. Let's turn it off. Let's erase all of this. So you had a ball of cells. The question is, the researcher asked a long time ago, you have a ball of cells. These ball of cells, which is called blastocyst, you remember that? We talked about it. And they all have nucleus. All of these cells have nucleus. Okay, so this is called, in case of human, blastocyst. Okay, the question was asked, how do these cells become head of a person, and these cells become leg? of a person. Do I make sense? A little bit? That's the next few PowerPoints. The next few PowerPoints is talking about what becomes the head, the anterior portion of the animal, and what becomes leg or arms, and so on and so forth. So that's the pattern. They call it setting up body plan. Setting up body plan. Body plan, head, leg. Or in case of the fruit fly that we are going to discuss here in a minute, we're talking about head and abdomen. Okay, animals, in case of insects, the fruit fly, they have a head, they have thorax, which means the chest cavity, and they have abdomen. Okay. They have abdomen. Okay, so let's go over that quickly. The last section of this chapter. Pattern formation is a development of spatial organization of the tissue and organs. In animals, pattern formation begins with establishment of major axes. The major axes are your head, thorax, in this case, head, thorax, and abdomen. That's you're talking about that. In case of us, our head, our abdominal, our thoracic cavity, our abdominal cavity, and of course our extremities. These are called extremities. Okay, that would be the body pattern. We have in animal kingdom, we have about 30 different body pattern. About that 30, 35. Okay, body pattern. In case of the arthropod, is different than us. For arthropods, it's different than us. We have head, thorax, as I said, abdomen, and then we have extremities. Our legs and our arms called extremities. But anyhow, that's the axis, major axes. Positional information, the molecular cues that control pattern formation tells a cell location relative to the body axis and neighboring cells. Oh, okay. In case of Drosophila, the fruit fly, this is the scientific name of the genus for fruit fly. Cytoplasmic determinants in the unfertilized egg determines the axis before the fertilization. And I'll show you a picture in a minute. And when you take zoology, we'll talk about the vegetative pole and animal pole, and based on that is determined if the animal is spiral cleavage or radial cleavage. Do not worry about whatever I'm saying right now, because when you get to zoology, we'll talk about it. But that's pretty much, that's what they mean. Eggs determine the axis before fertilization. And after fertilization, the embryo develops into a segmented larva with 3 larval stages. That's what he's talking about right here. So you do have an egg. This is supposed to be an unfertilized egg. This is the egg, and these are the nurse cells. These give food and nutrient to the egg cell. And then what will happen is all of these nurse cells are reduced, still unfertilized egg. We're still talking about unfertilized egg. They become smaller and smaller, and then the egg that has the genetic material and the mitochondria and other goodies becomes large. And after fertilization, what happens, of course, all of the nurse cells are gone. After fertilization, the cells undergo mitosis, you learned that. Then you start having the body plan, the head, thorax, and abdomen. Okay, the question is, that's what this section is all about. How do some cells recognize this is the head, this is the cells, differentiate, become cells of the head, and the cells differentiate, become cells of thorax, the chest cap area, and the cells differentiate, become abdomen. Okay, that's the question. And they answered it back in 1930s. Okay, so here is the actual fruit fly. You have a head, thorax, and abdomen right here. And of course, that's the picture right here. Let's go over it, how we can answer that question. Genetic analysis of early development of scientific inquiry. Edward Lewis and Christian Nelson. Do not worry about the scientist's name. I'm not going to ask. They won the 1995, they won the Nobel Prize, but most of the work has been done in 30s, 40s. Decoding pattern formation of Drosophila. Lewis discovered the homeotic genes which controlled pattern formation in late embryo, larva, and adult stage. So we are saying that in here there is a gene called homeotic gene, The homeotic gene is controlling the cells of what is becoming a head, what is becoming a thorax, and what is becoming an abdomen. That's it. Okay, so there is a gene. And the name of that gene is homeotic genes. So what is homeotic genes? Homeotic genes are genes that tell where the body plan goes. I wrote it down for you in red. So such as legs, arms, head, so on and so forth. Okay. Here is an example. They were able, this is the wild type, this is the normal. Whenever we say wild type, whenever for the rest of your life you hear wild type, it means that is normal. This is a normal head of a fruit fly. If you look at it, you have two eyes and you have the antenna coming out from right here. You have the mouth, everything. This is a normal type, but they, in the laboratory, they were able to mutate these homeotic genes. Okay, they were able to manipulate them in the lab, the genes, put some kind of chemical environment, ether, chloroform. They were able to manipulate those homeotic genes, and guess what happened? Right here, in the eye, you're seeing the legs. In case of the antenna, right next to the eye, you see the legs are growing because they mutated. Normally, animals do not do that. Okay, you know the eggs does not come out from the eye, but the legs are coming out of the eye instead of the antenna because they mutated those genes in the laboratory. And that's how they found it. How they found about homeotic genes. They are the genes that control the body plans. Those genes tell, hey, you are not supposed to grow here, you're supposed to grow the legs right here. Antenna is supposed to grow here. But if you mutate that genes, you have antenna coming, you have legs coming out of the eye. Okay, so anyhow. Axis establishment, maternal effect genes encode cytoplasmic determinants that initially establish the axis of body of Drosophila. These maternal effects genes are also called egg polarity genes because they control orientation of the egg and consequently the fly. Again, we'll talk about the polarity of genes. Your textbook is not going into details, of course, the chapter would be too long here, but in zoology we'll talk about it. This is an egg, this is an egg. We call this an animal pole, and then we call this a vegetative pole. And that is explained in next semester zoology if you're taking it. So again, I'm not going to even talk about that too much in zoology, but that's what they're talking about, the polarity genes. These are polarity genes that determine which portion of the egg becomes animal and which portion of the egg becomes vegetative. Bicoid, I gave you the MyoD gene, that was the name of a gene. Bicoid is the name of another gene. Okay, it's just the name of a gene. And what is the function of that gene? Morphogen that determines head structures. One maternal effect gene, the bicoid gene, affects the front half of the body. The embryo whose mother has no functional bicoid gene lacks the front half of the body and has duplicate posterior structure at both ends. Look at this. This is the normal, this is the wild type, if you would. You have the animal has a head region and a tail. This is the embryonic stage. This is the larval stage. Maybe you're familiar with the larva, the term larva. This is the larval stage. And you have a head and you have a tail. In the laboratory, they are able to manipulate those genes, okay, the bicoid gene. And when they manipulate it in the laboratory, then you have a tail here and you also have a tail in the larval stage. Of course, this never becomes a true animal. This never grows to become a fruit fly. Okay, when you manipulate those genes, then you have a tail here and you have a tail here.
Why do they do that? Just because?
To determine, they do these in the lab to determine what genes, like this one, to find that gene, the bicoid gene or MyoD gene. That's why they do that in the laboratory to determine what genes are responsible for what.
So then
they mutate it. Huh?
So now they know?
Now they know.
Oh.
they sacrifice a few fruit fly or mice, whatever, what have you, and then they know the genes, and then they can apply that to human as well. So they know there is a gene in human called bicoid gene that is responsible for our growth of the head, for example. Okay. Right here, what did I say? Bicoid, a morphogen that determines the head structure. An embryo whose mother has no functional bicoid gene lacks the front end. Again, when I was moving the slides around, the wild type here was up here, got stuck here, and the mutant one got stuck here. But that's why I showed it to you right here. This is a wild type. You have a head and tail. Oh, I did that slide because I wanted to put the information here. An embryo whose mother has no functional bicoid gene. This embryo, they blocked the bicoid gene, and that's what we got. That's what they've got. So here is a wild type, normal head, normal tail, and here is a mutant type. Tail, tail. They mutated the bicoid gene. The bicoid gene was not expressing itself, if you would. So there was tail growing from that end. Here is a bicoid. Again, you have the results. You have a bicoid mRNA in mature unfertilized egg. And then after, this is the cartoon picture. This is the real picture. That's the difference between these two. This is the cartoon drawing, if you would, and this is the real one. Egg. And then when the fertilization occurs, then the anterior end of the animal, that's what I was talking about, they call it animal pole, and this end they call it a vegetative low bicoid region, they call it vegetative pole. When we get to zoology, we'll just briefly talk about it. Bicoid protein in early embryo. Right here you have bicoid protein, here more of them and less of them right here. So you have the head of the animal and the tail of the animal.
Over the last few years, research has shown that mRNA molecules in the unfertilized egg of Drosophila determine the embryonic axes and future body plan of the fruit fly. The mRNA molecule that specifies the anterior-posterior head-tail axis of the embryo is transcribed from an egg polarity gene which codes for a protein called bicoid. This gene is active, mRNA is made, in the nurse cells that surround the developing Drosophila egg. The bicoid mRNA, once transcribed, is deposited into the Drosophila egg asymmetrically. More bicoid mRNA is deposited at one end of the egg than the other. Fertilization of the egg triggers the translation of the maternal bicoid mRNA into protein. The bicoid protein then diffuses through the egg, forming a gradient. The bicoid protein gradient establishes the polarity of the embryo along the anterior-posterior axis. The head and thorax will develop in the zones of high bicoid concentration. The abdomen will develop in the region of low bicoid concentration.
Okay, that's what I said. The polarity. They're talking about polarity. So the animal pole, he did not give it a high polarity. He just gave it in there. We give it animal pole is where the head and thorax grow, and vegetative pole is where the abdomen grows. Right here, he was talking about that. Okay, and you saw the protein molecules, the green dots. Those green dots were protein molecules that eventually become the head and thorax. Okay. The bicoid research was groundbreaking of three reasons. It identified a specific protein required for some early steps in pattern formation. Yes, he called it bicoid protein molecules. It increased understanding of the mother's roles in embryo development, and also it demonstrated a key developmental concept that a gradient of molecules can determine polarity and positions in the embryo. Oh, more. Okay, with that, let's move on to a little bit to the cancer. All of that research that you're wondering about, Reno, that you asked, well, all of that research helps you to understand cancer a little bit more. Okay, so that's, we work toward the application of all of these things. there is an application. Application of that is oncogenes. Oncogenes are the genes, they call them oncogenes, that are causing cancer genes in some type of viruses. So we do have oncogenes. Oncogenes are cancer-causing genes in some type of viruses, and viruses can transmit those to our cells. Proto-oncogenes are the corresponding normal cellular genes that are responsible for normal cell growth and division. Conversions of proto-oncogenes to oncogenes can lead to abnormal stimulation of the cell types. So if you have a cell you have a proto-oncogenes become oncogenes, and oncogenes, they are responsible for cancer. That's all I'm saying. Proto-oncogenes becomes oncogenes, and oncogenes, of course, can cause cancers. The cells can become cancer. Now, do we have other genes that can suppress this? We have other genes, they call them suppressor genes. Suppressor genes can block oncogenes. So tumor suppressor genes normally help prevent uncontrolled cell growth. Mutation that decrease proteins products of tumor suppressor genes may contribute to cancer onset. Tumor suppressor proteins, one of them is repair damaged DNA, control cell adhesions, and of course these are the things you learn in exam number 2 material. The adhesions act in a cell signaling pathway that inhibit the cell cycle. Interference with normal cell signaling pathway mutations in the RAS, or the name of the gene that I gave right here is a RAS. The suppressor is a RAS. Okay, the name of that gene is a RAS, proto-oncogene, and P53, another one, P53. These two are your suppressors. Tumor suppressors are common in human cancers. Mutations in RAS, or P53, lead to production of hyperactive RAS proteins and increased cell division. Suppression of the cell cycle can be important in causes of damage to cells. DNA P53 prevents a cell from passing on mutations to damage of DNA. Mutation in P53 genes prevents suppression of the cell cycle. So I remember the day that this came to the news, P53, it came to the news, everybody was jumping up and down, being happy. They found a cure for cancer. They found Amir, they found the cure for cancer when P53 was discovered. And that was happening during my lifetime. I've seen some of these things that is happening during my lifetime. You guys don't remember it. But that's not the case. We still do not have a cure for cancer yet. Okay, but the gene is that's supposed to prevent cancer is the P53 gene. They call it P53. Here is a good picture. I like it. It's kind of nice. Here's ultraviolet light, sunlight. What causes it? Causes damage. It damages DNA. Our DNA always are damaged. Now by viruses, by disease, by whatever comes across, sunlight, chemicals, you name it, our DNA get damaged. No matter what. By driving, whatever is environmental factors cause our DNA to be damaged. Then what happens, there is a protein kinase, a sequence of proteins are made to fix that damage, to control it, in a sense. Okay, so protein kinase eventually lead to synthesis of active P53, that's a P53 protein molecules, and it will go ahead and fix that transcription that is going to be made, the DNA is going to be fixed, P53, and then what happens? Protein that inhibit the cell cycle. So the cell cycle will be inhibited here when this is the normal situation, when P53 is being active, being synthesized, being made, then everything is going to be normal. Am I making some sense? The stop sign here, it means no cancer. That's what the stop sign means. And why no cancer? Because P53 is doing its job. It is fixing the DNA. I hope I'm making some sense. Okay. Now, what is the case that if this guy is damaged? Imagine who is protecting us in here? Policemen. If something happens, we pick up the phone and call 911. The police will come and protect us, right? What if the police is damaged? Who is going to protect us? Nothing. Reno go haywire. He goes chaotic. Brian will, start speeding me in the head, for example. Cancer will occur. Same thing here. That's the case here. So the ultraviolet light causes DNA to be damaged. These protein kinase are being made. And then what happens is some kind of mutation in the gene that is responsible for P53. So P53 is being made, but it's not being made like this. There's a mutation. And we talked about mutation in here in the past. There are different types of mutation. Okay, so if there's mutation here, it cannot fix the DNA, then nothing. Inhibitory proteins absent. Inhibitory proteins absent, and that leads to cancer. That's all I'm saying about P53. Okay. Now let's look at RAS. The multi-steps model of cancer development. Multiple mutations are generally needed. That's right. Not just one mutation will cause this full-fledged cancer. Thus, the incidence increase with age. As we age, the rate of the cancer can go up high. Again, at the DNA level, the cancer cells is usually characterized by at least one active oncogene and mutation of several tumor-suppressor genes. Okay, so these two are important, and I make them bold and I made them red for you guys to. It's just not one thing that causes cancer; it's multiple things. Okay, then let's talk about these two genes. So at the end of this chapter, and I will talk about these two genes. At the end of these chapters, there are a few genes that you learn. It's just nothing. The regulations of the genes, it's the name of the genes you learn. MyoD, bicoid, BRCA, BRCA1, and BRCA2. These are the genes that are responsible for breast cancer. Okay, so mutations in BRCA1, BRCA1, and BRCA2 are found in at least half of inherited breast cancers, and tests using DNA sequencing can detect these mutations. And then, Julian, what was her name? Angelino Giuliano, I'm sorry. Angelino Giuliano, sorry about that. She detected these genes in herself, and she was proactive. She went ahead and removed both of her breasts. Okay, so that's a lifestyle we choose. Here they are in case of colon cancer. This one is a RAS. I hope, yes, it's about RAS oncogenes. So let's go over normal colon epithelial cells right here. Loss of tumor-suppressor gene APC or other genes, and then colon wall, and then you have benign. What is benign? You can refresh my memory. Benign means what? It's not dangerous. Thank you. Benign, it means it's not dangerous. So you have a benign, you have a little polyp. They call this a polyp. Polyp forms right here, growth in the polyp. Activation of a RAS oncogene and loss of tumor suppressor gene SMAD4. Do not worry about this one, SMAD4, but worry about the RAS. RAS is a suppressor gene. It suppresses the genes that are growing to become cancer. And then larger benign growth adenoma, this becomes bigger, and loss of tumor suppressor gene P53. That was the last PowerPoint, loss of P53. And then what happens, additional mutations and malignant tumor, which is cancerous. You have abnormal growth. The role of viruses in cancer, the last slide, I guess, for this chapter, a number of tumor viruses, which that's the next thing I'm going to talk about, can also cause cancer in human and animals. Viruses can interfere with normal gene regulations in several ways. If they integrated into the DNA of the cell, that's what we're going to talk about next. Viruses are powerful biological agents can cause cancer. The next few slides in this chapter are they are review slides. You already